asks you, what is a gene? What will you say?
It is a segment of DNA containing the ORF
Right.
that
So
can be
forget about
transcribed and
ORF. Yeah. Yeah. Forget about ORF for now and just tell me without using the word ORF, what do you understand? It is a segment of DNA. That's correct. So we both understand and we are on the same page with respect to understanding that it is a segment of a DNA, correct?
Yes, sir.
Now there is a problem with this, just calling it a segment of DNA. What is the problem? Do you realize what is the problem? If I call gene as a segment of DNA, what is the problem with this? We can make any fragment of any segment of DNA I can cut, right?
Yes, sir.
Cut DNA anywhere. And then the next question is, will any segment of the DNA can be called a gene? Or is gene something special or is more defined? That is the next question, correct?
Yes, sir.
So that's why I told you that to call something a gene, it's not like randomly you put your, if I give you a representation of a long DNA in case of a long, in shape of a long thread, you keep your finger anywhere and from anywhere to anywhere, and will that be called a gene? No. So gene has some specific beginning and an end, okay, mostly. So gene starts, the coding of the gene starts with a start codon. Codons are just a pair of, not pair, but a group of three codes, three base pairs together. They are always read as three together, okay? In group of three, triplets we call it. So codon is a triplet of base pairs, and the start codon is one. There is only one start codon, which is AUG. So every gene starts with this start codon, okay? And then there are three stop codons where it should end with either UAA, UAG, or UGA. So start codon codes for methionine. This is the amino acid it codes for. There are 20 amino acids. You understand that?
Yes, sir.
Yeah. And therefore, when we define a gene, we define a reading frame, an open reading frame. Open reading frame is that region which starts with a start codon and ends with a stop codon. And in between, there is this coding region. So this codes for the protein. It's like how you start a English sentence. You always start with a capital letter, right? Like this is my space. And then you end with a full stop. So there are some rules, right? This can be called a sentence, right?
Yes,
If
sir.
I say is my, is this a sentence? Does this
No,
make any
sir.
sense? It's not a sentence. It does not make any sense. Okay. So it's not following the rules. Similarly, to define a gene, we have to define an open reading frame. So it is called an open reading frame because it has a start codon, a stop codon, and a coding region in between. Is that clear, Rabia?
Yes, sir.
Okay. So now can you tell me the complete definition of a gene and also if you understand it?
Genes are the segments of DNA containing an open reading frame that can be transcribed and translated into a functional protein.
Mhm. Perfect. And do you understand it thoroughly now?
Yes, sir.
Great. So would you like to give a quick recap from like this part, Mendel, why he chose P and what we did in the last class?
Yes, sir. So,
Yeah. Okay, quickly. Good.
yeah, so we discussed about Mendel. He was the father of genetics. His full name was Gregor Johann Mendel. And then we discussed that he chose a pea plant for the experiment, which is also called Pisum sativum. And then we wrote a question why we chose Pisum sativum. So the reasons are there are variety of traits to study. Second, it is easy to grow. Third, it has a short life cycle. Fourth, it produces a large number of seeds. And fifth, all traits have contrasting character. Then we discussed that in pea plants, there are seven contrasting characters. First one is height. Second one is seed color. Third one is flower color. Fourth one is seed shape. Fifth one is pod color. Sixth one is pod shape. And
Mhm.
seventh one is the position of the flower. And we also discussed the last thing in genetic parents is called the F naught.
Right.
And Mendel calls the gene factors before it was
Right. He did not, yeah, he did not know about gene and also, yeah. Okay. And what did Mendel do? We discussed a little bit about what Mendel did and all, right?
yes, sir.
What did he do?
He crossed three plants, which was one tall and one dwarf,
Mhm.
and then it gave rise to a tall plant, which was F1,
Yeah, so he
and
crossed
then
a tall and a short, tall and a dwarf, and he found that interestingly, in the first generation, all were tall, right? Now, this is not something you would expect when you are doing it for the first time, right? And then Mendel was also not expecting it, but then when he got it, maybe he thought that, okay, I did something wrong. So he repeated it. And he repeatedly got the same result. Now, this led him to propose this idea of two alleles where one is always expressed in the presence of other. Does it make sense to you?
yes, sir.
And therefore, let's start with today's class. And therefore, this idea led to formulation of certain laws. Now, Mendel, these are called laws of Mendel, laws of inheritance given by Mendel. But when Mendel was working on when he published or recorded his data, he did not actually assorted all these as laws. He just reported findings. Okay. His work was mostly for a very large, for a very long time, a large portion of his work, almost all his work, was non-recognized. It was neither recognized or appreciated by biologists, other biologists working. There were few reasons for it, and therefore this question comes in boards as well. That why did Mendel's work? You can write this question down. Why did Mendel's work go unnoticed for many years, for decades actually? Okay. This question can also be asked in another ways, like why his work was ignored, could not get a public popularity, etc., etc. But basically, what they're asking is, if someone did such elaborate and detailed work about genetics, why do you think it did not got popular or was not noticed? So we have Mariam joining in. Hello, Mariam, how are you?
Good evening.
Good evening, good evening. So Rabia just gave a quick summary of what we did in the last class, and now we are discussing uh, why the work that was done by Mendel was not popular or was not noticed earlier by many scientists. For many years, it was not recognized. So what do you think could be the reason? So some some biologists were not ready to accept what he did. Can you think of some reasons why it might be so? First thing, why do things, reach news or any work reach the public very fast these days? Why is communication very easy these days?
because of social media, I mean.
Yeah, social media. So what is social media? It is a way through which we can communicate using internet, right?
Yes,
So
sir.
internet has revolutionized the whole communication thing. So earlier, whenever scientists used to find something for many years, it would not reach every nook and corner of the world. It will be figured out slowly and slowly. When that scientist or their work will reach different countries, someone in that country will talk about it, or they will go and talk about it in some meetings, conferences, or when they will meet other scientists, etc. So communication was limited by physical reach or of the scientist or their papers or the letters they write to other scientists, like that. Okay. There was no internet where they could do these things and publish their data, and then everyone can just browse and see. Okay. So one major reason for which for a very long time it went unknown, it was unnoticed, is lack of faster mode of communication, like the internet. It was not there. Make sense?
Yes, sir.
But even then, let's say it reached some people heard of the work, it still was not accepted. Now, what can you think could be the reason? Why do people don't accept something?
Maybe his theory had some errors.
Maybe his theory had some errors. Okay, it had some limitations and some exceptions, but what he said was correct. That's why we are studying it right now. So his theory was not wrong. It didn't had errors. It has some limitations. It was not applicable to everything. But whatever for peas and for many other organisms, even for human, in many conditions, it is applicable. Okay, exceptions, about the exceptions, we just figured out the exceptions later, very late, when the work was tried to be repeated by other scientists. But when you hear of something and you just don't accept it, why? Why did that happen? What lead to people not accepting something? Because they don't understand it, right? You will not accept something if you will not understand it properly, correct?
Yes, sir.
Yeah. So one another reason was that Mendel, for the very first time, tried to use mathematics ratios to explain a biological phenomena. And this practice was not very common in back in those days. Mathematics and biology was very, very secluded, not like now that we know that everything, science is interdisciplinary. We whether we are studying biology, chemistry, physics, mathematics, we need all the fundamentals to do good science. Back, I discussed that back in time, it was not like that. Okay, chemists used to do reactions in their chemistry labs, laboratories. They used to work with chemicals. Biologists used to observe life, report what's happening in life, etc., etc. Mathematicians used to solve complex equations, numbers, and all. So he used, write down, so he used mathematics, concepts of mathematics to explain biological phenomena. Now, this was very, very, what you can say, radical, a thought back then. So people did not buy it. Biologists could not understand it completely, that what he's saying makes sense. He's talking about factors. They could not accept, because there was no proof that something like a factor exists inside cells that get inherited. And from a tall plant, if you cross it with a short, only the tall plant's factor gets inherited and expressed, etc., etc. There was no evidence, just observations and ratios. So biologists did not accepted it. Is it clear?
Yes, sir.
It was radical and not very well accepted by the biologists of that time. Anything else you think could be the reason?
Also, that Mendel was not a scientist at that time.
Sorry, Mariam, come again. Be a little loud.
It can be that Mendel was not scientist at that time.
Mendel was not scientist. So what do you mean by not scientist at that time? So
Like Mendel was scientist when he was proving these all things.
Mendel was scientist when he was.
So it's like he was monk, he was not scientist.
So when Mendel was there, back then there was no degrees given to call you scientist. Like people were not doing PhDs as degree and then they can be called scientists. Like now, scientist is a profession. You become a scientist after you do certain level of education, complete some education, complete research work, get a degree, work in a lab, prove something, publish, and become a scientist. Earlier, all these people were very curious researchers. So, but what you are saying, if I understand correct, you mean that Mendel did not have a big reputation as a biologist, right?
Yes, sir.
Yes, you're right. Because Mendel was not from the field. He was not like Aristotle, Darwin, or people who have explored biology from the beginning. He was not a naturalist, basically. He was a physicist by education, a physics student, a mathematician by his liking. He used to explain things through numbers and maths. And he was working as a priest in a church. And that's where, in that compound of the church, is where he did all his P experiments. So yes, he did not have much of a reputation as a geneticist or a biologist. That's correct. That is another reason when people figured out what he did. But that gets accommodated in this second point, that he explained biological phenomena through mathematics, which was very unpopular, uncommon at that time. So it was not very well accepted. Make sense? Does it make sense to you, Mariam?
Yes, sir.
So my point is clear, right? Your point is valid, but it fits in the second point. Let's merge it with the second point only. Okay. Any other reason you think Another reason was that a lot of things that Mendel recorded, he did not publish it as like papers. It was in his personal records and in the native language. So it was not in a universal language or published as papers, very widely circulated. So his reports, you can write, his study report was not widely circulated to reach a broader audience who could read his work and who could think about it and could find loopholes or repeat it and see if it works or not. So in science, this is a culture that whenever any scientist finds anything in the world, now it happens, it is called peer review. You have heard this word, peer review? Peer review system.
No, sir.
So you must have heard the word peer, right? Peer pressure. Peers are those who are at the same level as you are. So if you're in class 12, other 12s are your peers, right?
Yes, sir.
So peer review system is where in the scientific community, whenever you claim that you have found something, you have discovered something, you have invented something, you have to write a paper and that paper goes in peer review. So your other scientists will read that paper, will see the data, will read the results, and will decide and give their feedbacks that whether what they are claiming makes sense with the data they are presenting or not. Is it doable? Does the data is enough to support their hypothesis or not? If the review comments are in the favor of the paper, the manuscript, then only it is accepted. Okay, and other labs, so they have to, anyone who's publishing something or claiming to have discovered something, have to write each and everything in the protocol, in the method in detail, so that other labs can also try and test to replicate it. Okay, it's available for everyone. And only after a peer review, when people read and give their comments, oh, it makes sense, their data looks authentic, genuine, there is no fabrication, no cheating involved, and all the data that they have presented also supports the hypothesis, then only it is accepted as a published research in science. Okay, that's called peer review system. So this kind of system was not present back then. So people used to write opinions, people used to write and record observations. So not everyone was taken that seriously. Okay, only some big names were taken very seriously and were read, and their work was widely circulated. But others' work was not. And even Mendel was not that rich or had resources to publish and circulate his work widely because he was a priest. So make sense, Rabia and Mariam?
Yes, sir.
Yes, sir.
Yes. Now, after many years, after a few decades, Mendel's work was independently rediscovered. So whatever he wrote, it was kept in some archives. So the good culture about academics and literature is that earlier any piece of knowledge was regarded as a contribution to philosophy. So philosophy is the highest form of where you think about the source of knowledge or you think about knowledge itself. That's why the degree called PhD means doctorate of philosophy. In any field you do a PhD, you are basically a doctorate of philosophy of that field. Okay, so philosophers were regarded as with some respect, and their work was kept. Their opinions, articles, whatever are old early philosophers, right? Greek philosophers, Roman philosophers, even the Indian Vedic philosophers, the Islamic philosophers, whatever they wrote was kept somewhere and as archives. So even Mendel's work was there present, but it was independently rediscovered by 3 scientists. Okay, by first was Hugo de Vries, Carl Correns, and von Schermack. Okay, so these 3 independently, and remember this word, this is very important, they independently rediscovered his work, read the work, and in their different regions where they were present, and then they realized that what Mendel did was remarkable, because by that time people already started talking, biologists were talking to mathematicians, concepts of mathematics were being applied in biology. So when his work was rediscovered, they figured out, oh, it's amazing work. What he did makes a lot of sense. So then Hugo de Vries was the person who raised Mendel's work and made the laws. Okay, so he raised the value of Mendel's work to Mendel's laws, which we now read. Okay, so it was raised by this person. That see, Mendel did great work, and all his work can be summarized in 3 basic laws, which are called laws of Mendel, just like laws of Newton are there. Do you understand?
Yes, sir.
Yeah, so now let's talk about laws of inheritance by Mendel. So these laws were given by Mendel. Now, this story also tells you that, in science, you need selfless and curiosity-driven good people, right? Otherwise, if they were not good people, they would have rediscovered Mendel's work, but maybe decided not to give Mendel the credit, and then repeat the work and then say that, oh, I found this out. Yes or no? Because anyways,
Yes,
the
sir.
world was not knowing about it. But that's where we always say, even in peer reviews, it is said that science operates on trust. Anything that scientists do should have trust. Scientists should know that the world trusts them, the society trusts them. Okay, doctors, the work of doctors operate on trust. So when patients go and doctors say that, okay, now I'm going to give you anesthesia, the patient's life in completely in doctor's hand, right? Beyond that point, whatever doctor does, patient will not know anything about it. Yes or no?
Yes, sir.
So if you're choosing this profession of becoming a doctor or a scientist or a researcher, you are serving the humankind, and therefore you should have a lot of people should have a lot of faith and trust in you, and that's where you have to be selfless. You have to give credit to the person who deserves it, and this is done in science. Okay, so these laws, which were figured out, were as such. They were the following. So first law, about which we have already talked a bit in the last class, is called law of dominance. What does the word dominance means? Dominance. Dominance means domination, right? One dominates the other, correct?
Yes, sir.
So this says, write down, the law of dominance states that Law of dominance states that one allele expresses itself, one allele is capable of expressing itself one allele is capable of expressing itself over the other in a heterozygous condition. In a heterozygous condition. Heterozygous means, for example, capital T, small t. One allele for tall, one allele for short. That's called heterozygous condition. One allele is capable of expressing itself over the others in a heterozygous condition without any blending or mixing up. Without any blending or mixing up. Full stop. Such an allele is called dominant allele. Such an allele is called a dominant allele. Okay, and the other one is called recessive, right?
Yes, sir.
So it was very clear that like this was proven by Mendel by using multiple traits, but what he basically meant was this. Okay? This is called a heterozygous condition of alleles where this allele is always dominant and the other one is recessive. And the best example he found, the proof for this is when you cross a homozygous tall with a homozygous short. Okay, so this is for tall, this is for dwarf, and this is your F0 generation. Then in the F1 generation, all are tall, which is capital T and small t, right? We proved that above. Sorry, this one with a cross, with a Punnett square, right? So in the F1 generation, all are tall. This is called law of dominance. Okay. Now, this first law was working very well. It worked for Mendel for all the pea plants he worked with or did crosses with for a reason. You know, this is not a universally accepted law. This law has some exceptions. But Mendel could not find any exceptions because the 7 traits that he chose, all these 7, luckily show complete dominance. Okay, so they follow the law of dominance. So Mendel thought that every organism in the nature, every plant or animal will also follow this law of genetics. But that was not true. Okay, because when these other scientists started, remember I told you peer review. So when the law was rediscovered, the work was rediscovered and was given the status of laws, other people got very curious. They started working on it to find out, okay, does it also hold true for this plant or that plant or other animals, etc., etc. So that's where they found. So this kind of genetics now got a name called Mendelian genetics. What is it called? Mendelian genetics or anything that follows laws of Mendel, any gene, is called Mendelian inheritance. Is that okay? Does it make sense, people? Am I audible?
Yes, sir.
Yeah. So this is called Mendelian genetics. There is non-Mendelian inheritance as well, about which we will learn later in this chapter. But just write down that law of dominance is not a universally accepted law. It's not the universally accepted law in nature, which means it is applicable to 98% of things, but there are 2% exceptions, let's say, which do not follow this law, but still work, like still show inheritance pattern and all. Now, what are these exceptions? Anyone know? If this chapter has been discussed in your school, do you know about any exception of law of dominance of Mendel? First, tell me, is law of dominance clear to you? It is the first law.
Yes, sir.
Okay, so what do you think could be exception in nature? The law of dominance says that, see, in a heterozygous condition, one is always dominant over the other. So what could be exceptions to that? What will happen then that you will say that, oh, this is an exception? Tell me.
It says
Yeah.
incomplete dominance.
What does this mean? The word means. What does incomplete dominance means? That whatever Mendel said, yes, Rabia.
Yeah, it's like sometimes there was no dominant feature. Like it had one was equally dominant over the other.
So if you say equally dominant, it's a different thing. If you are saying incomplete dominance, it's a different thing. These two words means different thing. Two people are equally dominant means that there is still dominance, right? But incomplete dominance means that none of the allele is capable of expressing itself completely in the presence of other. Does it make sense?
Yes, sir.
Yes. So Rabia is completely right in saying that the first is, it's called incomplete dominance. It's very simple, right? Nature is just logic. If I claim that there is dominance whenever there are two alleles, then all you have to do, or all someone has to do, is to find a system where there are two alleles for a given gene. And when you keep it in a heterozygous condition, none of the allele is fully expressing itself. Okay. So that will just tell that my law is not universal. My law is not being followed. It might be getting followed 98 out of 100 times, but there are one or two exceptions where it is not getting followed. This does not tell that Mendel's law was incorrect. This does not, decrease the value of his work. This only tells us extensions, or what people found, additional knowledge that, yeah, his work is correct. This is how inheritance works. But there are systems in which this is not being followed. And in biology, let me tell you, there are very few fundamental rules that does not have exceptions. And those rules are so fundamental that life depends on it. Rest everything, almost majority of the things in biology has exceptions, because it is not about a story. It is not a story. It is not the story of a perfect creation. Biology is struggle, an evolution where life tries to find ways, different ways to exist. So you see all different kinds of life around you, plants, animals, microorganisms. You see viruses, you see bacteria. We are still struggling to decide whether a virus is a living or a non-living, whether it, what kind of arrangement it has, because viruses are not cells and they do not behave living outside the host body, but inside the host body, they show all signs of life. They replicate, they make their more copies. They also show metabolism, etc., inside the host cell. So it's very confusing, interesting and confusing both. So people are still trying to figure out how viruses evolved. Are they living or non-living? Are they at the border of life and death, like life and non-living material, etc.? So they are like an exception to a 5-kingdom classification. They do not fit anywhere in the 5-kingdom classification. Okay. Similarly, if I talk about any other thing, for example, you say that all living systems can see, can sense light, then it's not correct again. Plants can sense light, but not through eyes. Humans can sense light through eyes. There are many organisms that can sense light through different cells, which are light sensitive. But there are animals that do not sense light at all. They don't need to sense light because they do not interact with light. They live in very deep inside the ocean. So light is something that they do not encounter at all. So they have not evolved any senses to sense or pick up light. So you see, there are exceptions again, right? But some things are very fundamental. For example, metabolism. Now, metabolism is something which defines life. So this ground rule cannot be broken. If something does not show any metabolism, whether inside the host or outside the host, we will not consider it living. Okay. Every living system has to metabolize, make some chemicals inside itself to survive. Does this make sense to both of you?
Yes, sir.
So that's why biology has this reputation of being called the science of exceptions. But if you think properly, everywhere you find some exceptions, right? Even in physics and chemistry. But all we can say is it is more often found in biology because biology is about life. And life find all different ways to survive and flourish. That's why. So incomplete dominance was figured out in the flower color of Snapdragon. So the flower color of Snapdragon. Anyone knows the scientific name of Snapdragon? It's something and enter antirum antirum plant, something like that. Can you find it out from NCRT? What is the scientific name of Snapdragon plant?
And antirrhinium.
Antirrhinium, right?
Yes, sir.
Can you spell it?
ANTIRRHINUM.
Yeah, antirrhinium. Great. So the flower color of antirrhinium had two alleles for the gene. Now, what is color? How does color comes in biology? Like when you see any biological system that has a color, there are two ways colors can be found in biology. And talking about color, let me tell you a very interesting thing. How many of you have seen peacock feathers?
I've seen in videos.
Yes, Mariam, come again.
I've not seen in real, but I've seen in videos and photos.
Videos or images will also do. Rabia, you have seen it in real?
No, sir. Videos.
Videos, but peacock feathers are also like people also sell it. They should not, because for that many male peacocks are killed. But even if you've seen it in videos, what do you think of it? It's very colorful, right?
Yes, sir.
Anything special about peacock feather that you would like to tell me if you have seen in videos? Does peacock feather looks the same when you see it from any direction? Or does it look different? It shines and glows and the color slightly changes, right? Mostly it is bluish in texture, bluish greenish, dark greenish, or dark bluish indigo. But it also changes slightly, right? With light when we turn, twist it. Yes or no?
Yes, sir.
It's something like this, no? It's the feather is like this, but you will see some this kind of a patch, right? Which looks
Yes,
blue in the
sir.
center. And there are lots of small feathers, but this part glows different, this part glows different, right? So in biology, there are two ways to produce color, okay? And you will be surprised to know that we see a lot of things in biology, in a lot of living systems that are blue in color, right?
Yeah.
Butterflies are blue, fish are blue, birds are blue, their feathers are blue. A lot of things are blue in biology. But let me tell you, in biology, there are very few, just two or three found out till date organisms that can synthesize a blue pigment. So color in organisms can come either through pigments. For example, the human skin color comes from a pigment. You know the name of that pigment that gives human skin a color?
Melanin.
Melanin, right? So melanin is a pigment which is present in our skin cells, and the concentration of that pigment decides whether you are dark skinned or you are fair skinned. Okay, so that gives human colors. So similarly, there are other pigments that give other organisms the color. Okay, there are pigments that give carrot a color. It's called carotene. That's why the name comes from carrot. They have carotene as pigment. What gives plant a green color?
Chlorophyll.
Chlorophyll. Now, chlorophyll again is a pigment, right? But you might also have seen that there are other plant parts that are not green, right? In fact, all the leaves are not green. Have you seen any new leaf coming in a mango plant or a mango sapling growing or any other sapling growing? Not all plants, when they start growing, their leaves are green. Sometimes their leaves are reddish brown when they start. Then they became green after some time. Have you seen that happening in any plant, young plant?
Sir, I've seen mango plant.
Yes, yes. So clearly there is no chlorophyll or very less chlorophyll at that stage, but it looking brown or yellowish because of other pigments called xanthophylls. Okay, so there are xanthophylls, there are carotenoids, chlorophyll. Okay, all these are biological pigments. So one way color can come is pigments. The other way color can come is called the texture, the structure. So they are microstructures that trap light which falls on them and absorb most of the wavelength, but reflect only some. So most of the blue color in nature is a result of this phenomena, this physical phenomena, which is called diffraction, or which is called iridescence. It is not happening through pigments. So actually the peacock feather that you see blue is not blue. It has no blue pigment in it. Okay. It is just structured. If you look at that structure under microscope, it is just structured in a way that any light that falls on it, it does not allow all the light to reflect back. Only the blue wavelength reflects back, and hence we see it as blue. It absorbs all the other or traps all the other wavelengths. Okay. This is also the reason that the sky from Earth looks blue. It's not like the universe is blue in color. It has no color. The space has no color in it, right? But why sky looks blue is because blue scatters, the blue wavelength scatters the most because it is one of the smallest wavelengths. Okay, you know about Vibgyor, right? How do we, the visible spectrum is Vibgyor: violet, indigo, blue, green, yellow, orange, red. So beyond yellow, it is called the red shift, like towards the red. And before green, it is called the blue end. There is a blue end and there is a red end of the spectrum. Red is the longest wavelength, but the lowest energy. Blue is the smallest wavelength and the highest energy. So it scatters the most. Is that clear?
Yes, sir.
Some physics behind it. So very interestingly, scientists have found that there is one butterfly that produces a true blue pigment in nature. And if I found, I will share that paper with you in the next class. And it has a real blue pigment. Everything else that you see blue is just a physical phenomena. Isn't it interesting?
Yes, sir.
So I have a fish at my place. It's a Siamese fighter fish, betta fish, and it's blue. It's beautifully blue, blue with red patches, blue, red, and green, and all sorts of color it shows. But I just realized that it's all iridescent, right? Because that cannot produce blue pigment. There are very rare blue pigments in nature. Okay. Even some frogs look blue, right? Have you seen some frogs that look blue? Some humans have blue eyes. And I already told you that there is no blue pigment for eyes. There's only one pigment, and it is the concentration of that pigment that gives us that blue, green, grayish, brownish effect, okay, or blackish effect. Okay, so in this plant, let's come back to color. The color is because of pigments, okay? And there are two pigments responsible for this flower color. One is called the red pigment, another is a white pigment. Now, white is also a color. It is not like no color is called white. There has to be a white pigment to produce white color. If there is no pigment, it will be colorless, like a glass, okay, like water. So red color was because of a pigment, allele which gives red pigment, and let's call it R. White is because of an allele that gives white pigment, let's call it small r. So normally in this, if you find this genotype, capital R, it will be for sure red, like the flower is red flower, right? If this is present. Now this is a homozygous condition, correct?
Yes, sir.
In contrast, if you have another homozygous condition, small r and small r, you will have a white flower. Till here, everything is sorted. People are happy. Okay, that's great. That's what Mendel also told us. Now, the problem started here. If you have a gene or type which is capital R, small r, according to Mendel, what should be the flower color? If you would have asked Mendel, and if law of dominance would have been followed, what should be the flower color?
Red.
It should be red. But instead, this produces a different pink flowers. Now people were perplexed. Okay, wait a minute. This is not what Mendel told, because Mendel said that if you cross a tall plant with a dwarf plant, you will get all the tall plants, no intermediate height, no dwarf in the first progeny. Here, when they did that, let's do it. So when they did that, R crossed with small r, if you make a Punnett square, so these are the two gametes, same, these are the two gametes here. So all progenies will have capital R, small r, capital R, small r, capital R, small r, correct?
Yes,
This is
sir.
F0 and this is F1. So according to Mendel, all should be red only. But instead, all are pink. So what do you think is happening here? Rabia or Mariam?
The genes are mixing.
Sorry,
The colors are
gene,
mixing.
the alleles. The gene is one. The gene is for the flower color. It has two alleles, one on the one chromosome which came from father, one on the other chromosome which came from mother. And if it carries two different alleles to make a heterozygous condition, in this heterozygous condition, can I say that no allele is completely dominant over the other? If capital R was completely dominant, this would have been all red flowers. If small r was completely dominant, then this all would have been white flowers. But instead, both are blending in, right? There's a blending, or as Rabia used a word, a mixing effect, correct? Which means no one is completely dominant and there is a incomplete dominance in this case. Okay. Is it clear, everyone?
Yes, sir.
Yes.
Perfect. So when this phenomena was discovered in Snapdragon plant, it came out as a exception for Mendel's law of dominance. And now with increasing evidence, we have found that many genes in different organisms show incomplete dominance. Okay. Like in one organism, it could be a gene for color. But in Snapdragon, let me tell you why still Mendel's work is respected, because in Snapdragon, there are thousands of genes, right? 10,000, 15,000, let's say genes. So flower color gene shows incomplete dominance, but other genes do follow law of dominance. Okay. If you take a gene for the size of the leaf, let's say, as an example. So one gene will always be dominant over the other. So only one gene follows incomplete dominance, but other do follow the law of dominance. So do you understand what I mean to say? There will be some genes here and there in organisms that might not follow the full law of dominance. But it's not like all the genes in Snapdragon plant do not follow law of dominance. Okay.
Yes, sir.
Yeah. Okay. So that is one dominance. So write down in incomplete dominance. Let's write about it. What you have to write in exam. In incomplete dominance, no allele in a heterozygous condition, no allele in a heterozygous condition is able to express itself completely over the other. Completely over the other. As a result of which there is a blending of characters. There's a blending of characters. So this is one way through which law of dominance can be neglected or can have exceptions. Now, I think Rabia used another word. Can you think of any other way through which law of dominance can have an exception? What else can happen in nature and which once found, you can say that, oh, this is also an exception of law of dominance. One is incomplete dominance. What could be the other thing that can happen? Rabia gave a hint about it already. Rabia, Mariam? What else can happen? Come on. Either none of the alleles show a complete dominance over the other, or what could be the other scenario? The other scenario could be that both the alleles are equally dominant. Yes or no?
Yes, sir.
So law of dominance says that only one allele is dominant over the other. So one has to be dominant, the other has to be recessive. This is how nature works according to Mendel. But one we figured out that no one is dominant. The other is where both the alleles are equally dominant and equally express each other in the presence of the other one. So that's called codominance. This phenomena is also an exception to law of dominance. Incomplete dominance and codominance. So let's first define codominance. And in codominance, both the alleles in a heterozygous condition, okay? That is very important term, heterozygous condition. Both the alleles in a heterozygous condition are capable of expressing themselves equally. Are capable of expressing themselves equally. No one is dominant over the other. No one is dominant over the other. And both are expressed. And expressed equally. Make sense?
Yes, sir.
Now, anyone knows of any condition where codominance might exist? People? Okay, how many of you have seen? Okay. So both of you know about
Voice is
human. Sorry, yes, Mariam.
sir, I'm very clear.
Come again, come again. I could not catch you.
So your voice is breaking, sir. I'm not able to listen.
1 second. I think it's my issue. Rabia, can you understand Mariam clearly?
No, sir, for me, it's lagging. That's why.
It's the voice is a little muffled, Mariam. Come again. Be a little close to mic if you are not.
Sir, I was saying that your voice is lagging, sir.
No, I'm sorry. I'm so sorry, Mariam.
So
Yeah,
she's saying that your voice is lagging for her.
my voice is okay. Is my voice is also lagging for you, Rabia?
No, sir, it's clear for me.
Okay, then Mariam, I think it's your network because we can't understand you and you also can't understand me. If you want, would you like to reconnect? I can wait for a minute for you.
Okay, sir.
Okay, then let Mariam reconnect. Okay, Rabia, we'll wait for a minute.
Sure, sir.
Yeah. In the meanwhile, can you tell me if you know about any examples of codominance?
So I think
Any,
blood group.
you think blood group? Okay, that's great. Which one? Human blood group or animals blood group? Which one?
Human blood group.
Okay, that's great. So let Mariam join. I think she's joined. Okay, great. Mariam is back. Mariam, am I audible to you?
Yes, sir.
Perfect. Okay, you are also audible to me now. Yes. So when you were gone for a minute, I just asked Rabia if she have any idea or any example for codominance, and Rabia said human blood group is an example. Do you agree with Rabia? So can you elaborate what in a human blood group? What is a human blood group? How many different groups are there in human blood?
4.
4. Okay, what are they?
A blood group,
Positive.
B blood group, AB, and O.
Okay, once is A group, then you said there is B group, then there is O group. Okay, I should write group, otherwise it's looking like a positive or negative. O group, and what else?
AB blood group.
AB group. Okay, so where is codominance here? Okay, let's talk about how A group comes. So if a group is A, there are 2 ways it can have A group. So let's say this is 1 chromosome from father, 1 chromosome from mother. Okay, I. Either it contains A allele on both the chromosomes. So IA will give rise to A group. Is this codominance?
Yes, sir.
No, it is not codominance. This is the normal, no, like capital T and capital T. Remember the definition. The most crucial part of the definition is that in a heterozygous condition, they should co-express. That's why that is when it will be called a codominance. In a
Yes.
homozygous condition, then to Mendel also said this thing. Mendel said that if it is capital T and capital T, it will be tall for sure. And if it is small t and small t, it will be dwarf. But this is not a heterozygous condition. This is a homozygous. Both are same alleles. Does it make clear for you? I know these things are a little confusing. That's why I'm going slow. But we have to make a very strong foundation to understand genetics so that we never forget it. So is it a codominance condition, Mariam? Mariam, are you there? Is this IAIA where both chromosomes contain A allele? Can we call it a codominance? I'm not sure if Mariam can hear me. Rabia?
Yes, sir.
We cannot call it codominance, right?
Yes, I got confused.
Yeah. So what is the other way A group can come? If from mother you get A allele and from your father you get a O allele. You know, basically what does O allele means? Anyone has idea what is a O allele? A blood is called A group because A, there are 3 antigens. There is a A antigen. There is a B antigen. Sorry, there are 2 antigens. But there is nothing called a O antigen. O means no antigen, which means nil, absence of something. So there are only 2 antigens which can be present on the human RBC, A and B, according to this blood group. So human blood group, there are many ways to make groups. This is called the ABO grouping. ABO grouping system. Okay. in this ABO grouping system, there are two antigens that people take into consideration, A antigen and B. If someone's blood group is A, so they have A antigen on their RBCs. Okay. Either they get these antigens from both the parents or they get it from one parent. If from the other parent they are getting O, which means they are not getting any antigen. So A and O simply means only one antigen is present. So logically, whatever is present will be expressed. No competition. So is there a law of dominance or codominance here in the second situation? Yes, no? No, right? Because there is only one functional allele which is dominant over the other. So this is normal, just like law of dominance, no codominance, because this will be expressed. O cannot be expressed because O is nothing. O is a gene that give rise to no functional antigen. Does it make sense, Mariam and Rabia?
Yes, sir.
Perfect. Similarly, for a B group, there are two ways to be a B group person. Either you get IB and IB from both the parents, or you get IB and IO. Make sense?
Yes, sir.
What do you think will be situation for to become a O group? How can one become a O group person? Either you get there is only one possibility that you are a O group. When both your parents are O group. Does that make sense? Sorry. Sorry.
Present, sir.
No. Yeah. I understood what I said. So there's a mistake in my statement. When both your parents give you the O allele, okay? They can be A group, B group, or O group. For example, this parent, which is himself or herself, this. So the parent has a A blood group, but he or she has this one allele which they can give to one of the in one of their gametes, and from other parents, which is, let's say, B group, this allele can come, and hence the kid can be O group. Okay. So your parent could be anything, A, B, or O, and the kid can be O group, but there is only one possibility for the kid to be progeny to be O group, and that is that it gets IOIO from both the parents. Is that clear?
Yes, sir.
Yeah. Now, can a kid have O group when the parent is AB group? To answer that, first let's go to what it means to be a AB group. When can a person be AB group?
It has IA and IB.
Yes. When one parent give you A antigen, allele for A antigen, and the other parent give you allele for B antigen. This is the only condition when you can be you can be AB. Positive or negative, we'll talk about later, but AB blood group. Now, this is the scenario where we see codominance, right? Only this. This part of human blood group shows codominance because there are two alleles, one for A and one for B, and both are expressed equally in each other's presence. And if you see the RBC of these people on their RBCs, you will find one antigen which is A antigen and the other antigen which is B antigen. Then there is A antigen, then there is B antigen. And both the antigens are present throughout the surface of the RBC. So does this make sense that it is a codominance?
Yes, sir.
Yes. So write down very clearly that in the ABO blood grouping in humans, in the ABO blood grouping in humans the ABO blood grouping in humans, the AB blood group shows codominance of allele A and B. The AB blood group shows codominance. Not A group, not B group, not O group. Only AB group shows codominance. Is that clear?
Yes, sir.
Perfect. Now I have some hot question for you, high order thinking skill question. And these are the questions that come in entrance. Now you have to quickly tell me, if mother has a AB positive blood group and father has a O negative blood group, okay. Now in the progeny or in the child, which blood group is possible and which is not possible biologically? You don't have to answer in terms of positive and negative. Just tell me A, B, O, and AB. Which will be possible and which cannot be possible? Okay, let's go one by one. Is A group possible? Whether negative or positive does not matter. Is A group possible in the child?
Yes.
Yes. How? Mother can give A allele and father, of course, father has both the alleles. So father is this. This is father and this is mother. Mother can give A and father can give O, and it will become a condition like this. So the baby can have A blood group, correct?
Yes, sir.
Perfect. Now, can the baby be B group?
Yeah.
Think about it. Yes, same logic. Because mother is this, so mother can give this one, IB, and father is this, so he can give IO only. And hence baby will become this IBIO and a B group will be expressed. Perfect. Can the baby be AB group? Mariam Rabia, can the baby be AB group? Think about it. You have both the genotypes written here. Let me remove the positive negative here for sake of confusion. If mother is AB, father is O, does not matter positive or negative. Can a baby be AB group? No. It is not possible biologically. Can anyone tell me why it is not possible?
Because the alleles are from the mother only and not from the father.
Because father has no antigen allele. If mother gives A, the blood group of baby will become A. If mother will give B, it will become B because father can only give O. O means nil, no antigen. To become a AB, you need A from one parent and B from the other. Here, the other parent cannot give either A or B. So biologically, a mother with AB group and a father with O group cannot produce a AB group kid. If that is the case, then there is only one option. So they give you some such questions in entrance where they ask, so a mother of blood group AB and a father of blood group O have a 3-year-old son with a blood group AB. Which is the most likely phenomena? Option A, the blood group was decided by mother's allele. Option B, the blood group was decided by father's allele. Option C, the blood group could not be determined by any of the alleles. Option D, the kid was adopted. So the right answer is the kid was adopted. Biologically, it's not possible. Okay, now tell me, can the kid be of O group? If the father is O and the mother is AB? Okay, I want Mariam to answer this. This is the last thing we'll do for today. We will start exactly from here. I will give you more complex questions that have come in entrance on this topic. It's interesting in the next class. But Mariam, can the kid be O group in this situation?
So no.
No? Okay. Mariam says no. Why? Why?
Mother didn't.
Yes, because to be a O group, there is only one possibility, that both your alleles are IO. In this case, the mother is AB, so father will for sure give IO, but mother can never give another IO. She will either give A or B. In any case, either the baby will be A group or the baby will be B group, but the baby cannot be AB group or O group. Does that make sense, Mariam and Rabia?
Yes.
Yes, sir.
Perfect. So I want you, both of you, to go back and look at some more questions on blood grouping and what blood group is possible in the progeny. They also ask you back questions. If the child has this blood group, what potential blood group could be of the mother and the father? So try to find these kind of questions from internet, previous year's papers, and try to solve it. Okay. In the next
Okay,
class, we will
sir.
talk about the other two laws, and then we will finish Mendel's laws, and we will come to the inheritance part, the chromosomal part. Okay.